Yes, thank you very much, Mr. Chair and Co-Chair. Very thrilled to be here, very thrilled to present our work. At the same time, of course, I'm mindful that I'm one of the few remaining obstacles between you and the lunch break. Many of the presentations that we've heard have focused on the huge potential of stem cells, on all the positive aspects. I've actually come here as the spoiler. I want to talk about some of the aspects of cancer cells that actually they have a stemness or a stem similarity that are harmful. It's known that cancer metastasis is in part regulated by intrinsic characteristics of the cancer cells since the 1970s. And the debate has been on, well, what are these intrinsic characteristics? A lot of the work that we did in the 1990s was actually focusing on active homing. Right? Cancer metastasis has a pattern. It has an organ preference. And the thought was, yes, the metastasis genes actually make tumors migrate in a directed manner to their target organs. We were able to propose a mechanism based on one of the very common metastasis genes called osteopontin. Osteopontin is a cytokine in the immune system, but when it is active in cancers, it is actually a metastasis gene in just about 30 different kinds of malignancies. So we found that phosphorylated osteopontin can get cleaved roughly in the middle and generate two separate domains, one of which, the C-terminal end, binds to CD44 and makes cells chemotax. And the N-terminal end binds to integrin alpha V beta 3, the integrin we've heard about earlier today, and actually allows the cells to crawl. Haptotaxis is the movement of cells on a solid support. When the highest concentration of osteopontin is reached, the cells spread. The signal transduction changes, metalloproteinases are secreted, and invasion is facilitated. This has not been invalidated. But what we did do back then was to overestimate the importance of the directed migration and invasion. We know now that the actual major limiting factor for metastasis is anchorage-independent survival. Take any healthy epithelial cell and detach it from its microenvironment, detach it from its substratum, and it will undergo a form of cell death called anoikis. So there is a redox problem and an energy problem in these cells, and these cells will die. Exempt are hematopoietic cells and tissue-resident precursor cells. Once there is tissue repair needed, once there is tissue damage, the precursor cells can be mobilized without dying. Then we know that malignant tumors, even at their very early stages, even when they are very small, already shed tumor cells into the circulation in large numbers. 99.99% of those cells die in the circulation before they can reach their site of implantation. So fortunately for us, we're dealing only with a problem of 0.01%. But those 0.01%, when they come through, can become lethal. Here's what we know about anoikis. Differentiated, untransformed, non-hematopoietic cells cannot survive once they lose the contact with their environment. So it's really our blood cells that are enabled to do that, and our tissue-resident precursor cells in the context of repair that are able to actually survive. All other cells die from anoikis. We know that anchorage-independent survival, therefore, is an absolute necessity for cancer cells. So here's the sequence. Epithelial cell detachment leads to a loss of glucose transport, and that will turn out to be key. A loss of glucose transport causes an ATP deficiency, and the energy deprivation then leads to programmed cell death. So what we have in cancer cells during invasion is an upregulation of the energy metabolism. We have an upregulation of mitochondrial activity, an activation of oxygen consumption, and ATP production. And I want to show you the data on a new function of osteopontin. The cytokine that we had previously associated with migration and invasion actually also is very important in this process of supporting anchorage independence. And you see that I'm referring specifically here to a particular splice variant. So what is that osteopontin? Osteopontin is a Th1 cytokine secreted by T cells and macrophages. Its full-length form, actually the exons are correctly labeled. Exon 1 is untranslated. Full-length form has 6 translated exons and stimulates the immune system. Now, we rediscovered splice variants that had been described to exist, but nobody knew what they were doing, that are actually present only in cancers. They have not been described in any healthy cell yet. So Splice variant osteopontin C loses exon 4. Exon 4 is fairly big, so it's the shortest splice variant. And osteopontin B loses exon 5. That's the intermediary-sized variant. If you take breast epithelial cells, these are non-invasive immortalized cells. You culture them in soft agar. That's a way of testing their anchorage independence. They form very moderately sized clusters that we call clones. Transfect these cells with osteopontin A, the full-length wild type, and you see that the clusters are roughly intermediate. Transfect them with osteopontin C, and not only are these clusters huge, they actually form these protrusions that we believe reflect the rapid expansion and the stretching out into the soft agar. The secreted versions of these cytokines are needed because when we add a specific antibody, we can reverse the effect. So it's the classic of an autocrine mechanism. The cytokine is secreted, it binds back to the receptor on the cells and allows the anchorage independence. And yes, we see that reflected also in the ATP generation. As I had indicated, the ATP levels in resting cells are relatively low, and once you transfect them, with A, it's intermediate, and with C, it's higher. And if I may ask you, please make a note of the co-transfection and what is either an additive or a synergistic effect. We'll come back to that in a moment. Right now, let me focus just on what we found about osteopontin C. So osteopontin C was the primary supporter of anchorage independence. And back when we identified and first described that, we did a microarray analysis, and we identified a large number of oxidoreductases being induced by osteopontin. And actually, it turns out that what is listed here as electron transporters, many of these genes also have a function as oxidoreductase genes. That was actually not all that unexpected. It was actually not all that surprising. There had been a small but substantial literature that had already indicated that the generation of reactive oxygen intermediates is actually required for metastasis. So the production of some hydroxyl radical or superoxide anion or hydrogen peroxide is actually supportive of the dissemination of cancer cells rather than inhibiting it. Right? There are these pleomorphic functions of reactive oxygen intermediates in cancer. In terms of metastasis, the production of some level of reactive oxygen intermediates is actually needed, and this finding here was consistent. So what's the pathway? How does this support anchorage independence? Actually, I need to mention we also confirmed that in patient samples. So we have a very good correlation here with some of the oxidoreductases with osteopontin levels in patient samples. So what we had found in cell lines was confirmed in these patient samples. And actually, you notice here when I project the names of these oxidoreductases, these are mitochondrial chain molecules, and glutathione peroxidase is part of the hexose monophosphate shunt. So we did a metabolomic analysis, and we're surprised to find a few molecules that we had not really predicted. Creatine, glycine, glutamine, glutamate, and actually we will come back later to phosphoethanolamine and phosphocholine. Now, we have microarray, we have metabolomic analysis, and we have the database searches of some of the publicly deposited databases that at least showed a correlation with osteopontin levels in cancers. And from that, we were able to assemble a pathway that actually does, as we had expected, feed into ATP generation. All of the molecules that are boxed in red have experimental support as being upregulated by osteopontin or by osteopontin C. So you see that the mitochondrial respiratory chain on the bottom is supported, the tricarboxylic acid cycle that feeds into it, upstream glycolysis, and glycolysis being regulated by the hexose monophosphate shunt. In each and every segment of this pathway, you have the upregulation of some component feeding toward an increased amount of ATP. Also, a lateral interaction here is the glycerol phosphate shuttle. The glycerol phosphate shuttle that activates FAD and stimulates the conversion like in complex 2 to complex 3, accelerating the oxidative chain in the mitochondria. So this is one major connected pathway that is collectively induced by the multiple effects of osteopontin C and leads to ATP generation. There's the other component. As I had said, our metabolomics analysis was somewhat surprising in terms of what it identified. It did identify glycine and creatine. So glycine and creatine can actually also lead to ATP generation or ATP regeneration from ADP. We found phosphoethanolamine and phosphocholine elevated, and we believe that this is not functionally important. We believe this is a consequence of the metabolic changes, that actually by scavenging serine to feed into the glycine pathway, we are not able to convert phosphoethanolamine and choline into phosphatidylserine, and there's a backup and an accumulation of these molecules. So one of the key findings here is that osteopontin C supports anchorage independence by activating the energy metabolism. But we're missing something. There are two components that are missing. First, I had indicated that glucose deprivation is a component of anchorage independence. We haven't accounted for the glucose deprivation or the overcoming, the overcoming of glucose deprivation in metastatic cells with osteopontin C. And the other factor is that osteopontin C, our splice variant, is never expressed without osteopontin A. It's never there without the full-length form. So what does the full-length form do? Is it inert to metastasis? Is it anti-metastatic? Or does it actually synergize with osteopontin? With osteopontin C. So yes, we actually found that there is a synergistic effect between the full-length form and the splice variant. You see here a pattern that looks very similar to our ATP levels that we had measured earlier, and this is just the soft agar clone formation. When you have osteopontin A transfected, you get intermediary clones. Osteopontin C is more effective. Once you put them both in, we have something that looks like an additive, maybe a synergistic effect of A and C. So we did our metabolomics analysis on osteopontin A transfected cells, and we were very surprised by what we found. You see that the main metabolite that is altered actually is glucose. We had totally not expected that. We had totally not predicted to find this strong induction of the glucose levels. Now, from our previous analysis, from the microarray that we had done years back, we also identified STAT3 as upregulated by osteopontin A, and we actually did the experiments. There is a STAT3 inhibitor, a peptide that is cell permeable, that is commercially available that we used, and we found that yes, if we add the peptide in soft agar colony formation, the peptide does reduce the size of the soft agar clones in the osteopontin A transfectants, but not in the osteopontin C transfectants or in the vector. And confirmed that then with our transfection assay using the genetic approach, and we find the same pattern here, where the dominant negative actually inhibits osteopontin A transfected soft agar colony formation, but has no effect on the osteopontin C colony formation or on the vector. We are not surprised, given that we are dealing with metastases genes and not oncogenes, that the growth on regular plastic is not affected, right? This is complete medium, just proliferation on plastic is unaffected. Once again, these are metastases genes. These are not oncogenes that we're dealing with. So we took our dominant negative STAT3 and asked, well, what happens to our metabolites? And yes, we found that STAT3 actually is involved in that pathway and does affect a number of the metabolites that we had previously identified as upregulated by osteopontin A. So from all of our data, all of the various approaches that we had used, we inferred that there must be a pathway induced by osteopontin A, which includes glycerophosphocholin, leads to the activation of STAT3, and through that mechanism induces IGF-BP5 and ApoD. And these are classical regulators of glucose. They were known as regulators of glucose, just not in breast cancer cells. So there is a physiologic role that when it's absconded with by the cancer cell, turns into a pathophysiologic event. By having osteopontin A upregulate glucose, we now have our source. We now have our feeder into the osteopontin C pathway. So osteopontin C, never expressed without osteopontin A, utilizes the glucose and feeds it into the respiratory chain and hexose monophosphate shunt, and therefore generates the amount of ATP that is needed to overcome anoikis. So you see that we've started out with one of the osteopontins here. I'm talking about osteopontin A first, and we did identify STAT3, IGF-BP5, and apolipoprotein D, and all of those affect glucose homeostasis. This pathway was novel. We had never thought of metastases genes as affecting metabolism. Now we see that a metabolic component is actually important. And we see that metabolic skewing by the various splice variants actually is a facet that is needed in metastasis formation. You see that anchorage independence is largely guided by metabolic adjustment. The tumor cell that is in circulation will, to a large part, change its metabolism, upregulate ATP, and therefore become resistant to anoikis. So the autocrine secretion of osteopontin variants is an essential pro-metastatic pathway. So summarizing the previous slide just in one sentence, in anchorage-independent cancer cells, osteopontin A stimulates the generation of glucose, and that glucose is then utilized by osteopontin C for energy generation. So what have we learned of actually a number of years of working on this molecule? We realized that anchorage-independent survival and expansion are achieved through metabolic adjustment within the de-adherent cell. Pro-metastatic ligands adjust or skew that metabolism, and that is key for the anchorage-independent survival. And it turns out that distinct metabolic changes are induced by osteopontin A and by osteopontin C, and those two splice variants work together. The role of osteopontin A is glucose uptake, and utilization is then mediated by osteopontin C. Together, they satisfy this increased energy requirement of the de-adherent cell in circulation. And so we propose that metabolic responses to environmental cues are likely much more common in cell biology than has been recognized. Right of the signal transduction research of the 1980s was talking about what happens at the receptor level is transduced into the nucleus and changes gene expression. There's a lot of signal transduction that doesn't need to go into the nucleus, that actually is taken care of via other means, and a lot of the signal transduction used in metastasis is in fact epigenetic. And so we have now the metabolic component. Cancer research and cancer treatment has progressed quite a bit. Right, since 1945, the discovery of the first anti-cancer agents, via 1965, the combination chemotherapy, and then 1997 and 2001, the introduction of molecularly targeted therapies. But we're still lagging behind in treating metastases. So hopefully by elucidating the molecular mechanisms that are required for dissemination, we'll get one step closer to treating this aspect of cancer. Thank you. Am I? Most of the work that was done in MCF7 cells. MCF7 are not fully transformed. They are immortalized breast epithelial cells that do not metastasize in vivo. Of course, for publication purposes, you always have to confirm in other cell lines. CR75 is another one of those non-invasive transformed cell lines. Similar properties to MCF7. Those are the two cell lines that are osteopontin totally negative. It's very difficult to find cells in culture that are totally osteopontin devoid. Both of these cells are totally devoid of osteopontin, so transfection is over a clean background, is over a clean slate. This goes back to the very early days of signaling. We have ATP receptors on neutrophils that actually can utilize extracellular ATP for migration. And yes, I believe that maybe if ATP leaks out of the cells, that it could have this effect via those extracellular sensing mechanisms. So it's quite possibly ATP that has left the cell. I don't know about the intracellular ATP, but yes, of course, motility will require, right here on the right, motility will require a lot of energy to reassemble the actin cytoskeleton.